Amyotrophic lateral sclerosis, ALS, often called Lou Gehrig's disease, is a degenerative disease of both the upper and lower motor neurons. As more and more motor neurons become affected, they are unable to send signals for movement to the muscles. Loss of voluntary muscle movement and coordination develop. Over time, muscle weakness and spasticity develop. Unfortunately, there is no cure for ALS. Death from amyotrophic lateral sclerosis usually occurs within 3 to 5 years from diagnosis.
Unfortunately, the cause of this appalling disease has still not been determined by scientists, with no identified cause in 95% of ALS patients, a group regarded as sporadic ALS. However, in recent years, scientists have linked about 5% of cases to a family history of ALS known as FALS. Links have also been made to mutations in the copper-zinc superoxide dismutase gene in FALS, and since then, scientists have been making strides in linking mutation in this gene with several other instigators, ranging from the role of oxidative stress to impaired axonal transport. One of the key connections made in recent years was the ability for scientists to link the tdp-43 protein to amyotrophic lateral sclerosis. tdp-43 has served as a pathological hallmark of a broad range of neurodegenerative disorders for decades, ranging from Alzheimer's to recently established ALS. Normally, its location in the nucleus helps to mediate the storage of information required for making proteins needed for cell function. A lack or loss of this protein in the nucleus and initiated presence in the cytoplasm causes the accumulation of pathological cytoplasmic aggregates in the brain and spinal cords. Afterwards, accumulation of abnormal hypophosphorylated tdp-43 variants as a result of its location in the cytoplasm is then speculated to cause a loss in cellular function that derives from a loss of normal tdp-43, thereby impairing the capability of these already affected nerve cells to a further extent. tdp-43 also plays a major role in another neurodegenerative condition known as frontotemporal dementia, or FTD. FTD is characterized by gradual and progressive changes in behavior and personality, and like ALS, progressively gets worse. Pathological aggregation of the tau protein is noted in the condition as it is considered part of the tauopathy family, a class of neurodegenerative diseases. Tau represents one of the major hallmarks of diseases like FTD, and its pathological aggregation serves as an indicator of the disease mechanisms. Abnormal phosphorylation and misfolding cause the formation of these insoluble aggregates, and both of these have been linked to components within tdp-43. Both disorders in recent years have been linked together, and it has been determined that 20% of patients with ALS also develop FTD, and 15% of FTD patients develop ALS, a new classification of disorder known as FTLD-MND. Tau aggregation, tdp-43's prevalence in neuronal and glial inclusions, and abnormal phosphorylation in both diseases provide further evidence that the two disorders may share a common underlying mechanism, to the point that some scientists are hypothesizing on two ends of a disease spectrum. Many scientists have been observing both tdp-43 and tau expression regarding their role in neurodegenerative diseases. The Neurodegenerative Disease Laboratory at the Mayo Clinic does just that, focused on investigating cellular mechanisms that cause neurodegeneration in diseases characterized by abnormal protein aggregation, and highlighted on the role of tau and tdp-43. Their goals are aimed to gain insight on the process that initiate abnormal aggregation of tau and tdp-43 and the harmful consequences resulting from the formation of these aggregates. In a study by Peter Ash at the Mayo Clinic, he analyzed the structure and function of tdp-43 neurotoxicity in C. elegans. He engineered pan-neural expression of the human tdp-43 protein in C. elegans that exhibited an uncoordinated phenotype and abnormal motor neuron synapses, then investigated the contribution of specific tdp-43 domains. They were able to determine that tdp-43 neurotoxicity can result from either nuclear activity of the protein or accumulation of cytoplasmic aggregates, suggesting that the two different mechanisms of tdp-43 neurotoxicity. Possible treatment for alleviation have surfaced in regards to ALS-related disorders, including the more well-known riluzole treatment and tetracycline derivatives such as minocycline. Others have also noted the use of minocycline in other diseases in the same spectrum as ALS. Wendy Noble of King's College in London demonstrated how minocycline reduced the development of a normal tau species in models of Alzheimer's disease. Alzheimer's disease is characterized by the presence of neurofibrillary tangles of hypophosphorylated aggregated tau protein and deposits of amyloid beta peptide, serving as a prime example of the tauopathies. Minocycline, already noted for its neuroprotective effects, were used in several models of mice to determine its efficacy against tau pathology. Treatment of minocycline in transgenic mice resulted in reduced levels of tau phosphorylation and insoluble tau aggregates, and lowering generation of caspase-3 cleaved tau fragments. They concluded that minocycline has therapeutic promise for Alzheimer's disease and as a novel therapeutic agent for the treatment of related tauopathic disorders. The study will use 3 strains under the GAL4 UAS system. Before this is even begun, there must be solid experience in the ability to sex and cross fruit flies in an efficient manner, as well as familiarity established in the fruit fly protocols of feeding, maintenance, and care. The 2 strains under UAS control expressing the human tau GFP and tdp-43 characteristics will be sexed and crossed with the GAL4 strain expressed in the eye, to create 2 sets of strains that express human tdp-43 and tau in the eye of the fly. 2 treatments of 100 micromolar memantine and 200 micromolar minocycline hydrochloride will be inoculated into the food supply of the larvae and feeding for at least 15 minutes on the 3rd day after hatch, with the 50% drug concentration in the solution, but this will be subject to changes in the initial pilot studies. Then observations will be made over the next 3 weeks after adulthood is reached by both groups of tdp-43 and tau. The tdp-43 group will focus on the observation of necrotic lesions and ommatidia loss in the fly eye, as demonstrated in Lee's procedure in 2010. Measurements will be based on the levels of severity in the eye defects and the gradual progression in retinal degeneration over the course of 3 weeks. The tau-affected group will focus on the fluorescent GFP analysis of the stained eye with image J and the CTCF formula to observe age-onset degeneration to the eye and its parts. SPSS ANOVA and Chaffee post-hoc value of less than 0.05 will be used for statistical analysis.